Okay, hello everyone. Thank you very much for having me. My name is Chung and I'm from Ghent University Global Campus in Songdo, Korea. So today I will be talking about our new findings in MAP kinase signaling networks in Arabidopsis that I and my colleagues worked on for many years in Professor Jenshin's lab at Harvard Medical School. And also, furthermore, I also would like to talk about what I'm working on together with my students in my lab in Korea, which is also dissecting the MAP kinase signaling cascade for plant immunity and development. So as we know, every living organism constantly senses intrinsic and environmental signals and reprograms cellular responses to grow and maintain their lives. So the signaling process is achieved by various key signaling molecules, and amongst MAP kinase cascade is the most ancient and essential regulatory module I like to talk. So it's been found in all eukaryotic systems examined so far, not only in the single-cell system, but also in the multicellular organisms, including humans and plants. So after signal sensing, this three-tiered MAP kinase cascade composed of MAP triple kinase, MAP kinase, 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 and MAP double kinase, MAP kinase, kinase, and MAP kinase. These three-tiered protein kinase cascade is triggered to activate sequential phosphorylation relays to directly modulate substrate involved in very diverse biological processes. So the idea that MAP kinase regulates very broad spectrum of cellular responses has been enriched by very extensive biochemical and molecular analysis such as protein chip experiment and phosphoproteomics, and also a large-scale protein-protein interaction studies that have discovered physiological substrates of MAP kinase. So the identified MAP kinase substrate include proteins with very diverse functions, including various transcriptional factors and translational regulators, protein kinases and phosphatases, and other classes of proteins like some biosynthetic enzymes, suggesting the roles of MAP kinase in numerous cellular pathways. So far we have ample candidates modulated by MAP kinase. And we all know that MAP kinase is very important for regulating signaling pathways in multiple programs. So then the remaining question is, how can we define the function of MAP kinase precisely in a specific signaling pathway? So one can think that maybe we have Arabidopsis genome already sequenced, so we know which ones are the MAP kinase cascade genes in Arabidopsis by sequence homology prediction. And then if we have genetic mutants with phenotype, so MAP kinase genetic mutants with phenotype, so then we know what they do, we know what MAP kinase do. But that's the simple way, when you define the function of the genes. But however, the real world is not that simple. So mapping genes to their function is called genotype to phenotype problem, where phenotype is whatever changes in the organism when gene function is altered. But the relationship between genotype and phenotype is actually not always simple one-to-one mapping between genes and phenotype, because phenotypes are result from the interactions, multiple interactions associated with the specific genes. So therefore, in most cases, phenotypes are actually complex because more than one gene can contribute to the expression of the phenotype. And these interactions could be developmental stage specific and also environmental condition specific, and also like tissue or cell type specific, especially in case of multicellular organisms like humans or plants. Arabidopsis thaliana, as we have here. So therefore, it is very difficult to accurately assign the gene function from the phenotype. And you can imagine MAP kinase gene is actually, their function is like a master regulator of many pathways. So you can imagine that MAP kinase gene would have very complex networks from the numerous connections with other gene products. And then, these interactions can result in multiple phenotypes. So if one of the phenotype, for example, like this pathway, so if one of the phenotype, from one of the network branch is very dominant, so the phenotype itself is very dominant, therefore, if it masks or affects the expression of or observation of other phenotypes that are originated from other branches, it will be partially misleading us to know the details of the gene function. For example, actually in case of MPK3 and MPK6, they are in single genes, they are functionally redundant in most cases. So the single mutant of MPK3 or the single mutant of MPK6 does not have much phenotype. However, when you have a double knockout of MPK3 and MPK6, as you can see, they are embryo lethal. Okay? So when both of them are gone, the mutants becomes dead. So in case of MPK3 and MPK6, we don't even have a plant material to study the function of MPK3 or MPK6 in a specific pathway. And also in case of MPK4 mutant, especially when this mutant is grown in soil, as you can see, the plant is very severely dwarfed compared to wild type, very small size. And also in these small tiny plants, there is a very high activation of autoimmune response. So they constantly have expression of immune genes. So if we do some transcriptome analysis in these MPK4 mutants compared to wild type, you can actually find about 1/3 of the total genes in Arabidopsis. They are differentially expressed in MPK4 gene. So these tiny mutants already have accumulated the consequences of losing this MPK4 gene function in these mutants, by having like a very perturbed gene expression pattern. So we cannot actually use this plant material to study a specific function of MAP kinase 4 in any signaling pathways, because there is already like a secondary response, physiological consequences are accumulated in this mutant. So it's been a problem in the field of MAP kinase, not only in plants, also in other organisms, due to the limitations of MAP kinase genetic mutants. So then to overcome all these problems, what we can have is something like a toggle switch, so that we can precisely control the timing of MAP kinase activity during specific signaling pathway. So for example, in case of MAP kinase 4 genetic mutants, so like this one, so from the zygote stage, they have the loss of MPK4 function. So then as the plant grow, there is accumulation of the perturbation in the system. So we can really use this plant material to study the specific function of the genes using this plant. However, if we have like a switching system, so we can control the timing of MAP kinase activity, so we can just grow the plants, and then only when we want to inhibit the MAP kinase activity, so we can control the MAP kinase activity precisely, during any developmental stage or any time that we need it. So the breakthrough idea came out early 2000s, actually, like about like 20 years ago. So the idea is actually very simple. So here I'm showing the ATP binding pocket of any protein kinases. So as you know, every protein kinases accept ATP. So this is ATP molecule. And so as I said, this is ATP binding pocket. And this is the structurally critical amino acid residue for the pocket size. So in general, they are a large amino acid such as tyrosine or threonine. But if we mutate this large amino acid to a small one such as glycine, which is the smallest amino acid, so then this mutant kinase will have a relatively enlarged binding pocket because you have more space available in the pocket. So then in this mutant version of protein kinase, this ATP binding pocket not only can accept ATP itself, but also it can accept variants of ATP such as ATP analog, bulky ATP analog, which has one or sometimes two more benzyl ring on ATP molecule. So this type of ATP analog can only specifically bind to the mutant kinase, which has enlarged binding pocket, but this bulky ATP analog cannot bind to the wild-type kinase. So then this can function as an inhibitor of mutant kinase and inhibit the activity almost immediately. So we applied this system to Arabidopsis MAP kinase. So actually the approach that I just described was first developed in yeast system using cyclin-dependent kinase as a model. So then after the work was published, we applied this system to Arabidopsis MAP kinase. And so then we generated inducible MPK4 mutant and inducible MPK36 mutants in the background of MPK4 genetic mutants and in the background of MPK3 and MPK6 double mutants. So as you can see, this MPK4 genetic mutant is complemented with MPK4, which has a mutation on the gatekeeper residue, so changing tyrosine residue to glycine, and MPK36 double mutant was complemented with MPK6, changing tyrosine to glycine. So as you can see, the transgenic plants, they grow as normal as wild type, indicating that without a treatment of ATP analog, without treatment of inhibitor, this mutant kinase, they can perform their functions like wild type kinase. So plants can grow normal. But then importantly, so here I have an example of the phenotype of just MPK4 genetic mutants and MPK36, a genetic mutant, which is embryo lethal. So you can see that the mutant phenotype was complemented in these transgenic plants, but they are supposed to be inducible MPK4 and MPK36 mutants, which meaning that when we apply ATP inhibitor exogenously, there is an inhibition of MAP kinase function. So we check the phosphorylation of the physiological targets in these mutants. So as you can see, ACS6, WRK33, they are actually known substrates of MPK3 and 6, and MKS1 is the known substrate of MPK4. So as you can see, upon FLAG22, so this is a bacterial immune signal, after FLAG22 treatment, there is a phosphorylation of these substrate proteins by MAP kinase activity. However, when we applied FLAG22 together with ATP analogs, here 3-MBIP is a short name of ATP analog that we use, which has one benzyl ring on it. Then, so as you can see, when we applied ATP inhibitor together with the FLAG22, the phosphorylation pattern was abolished in these mutants. So this result indicating that we have a tool that we can control the activity of MAP kinase in vivo by exogenously applying ATP analog inhibitor. So having these tools ready, so we set out to answer long-standing questions in MAP kinase signaling in Arabidopsis. So there are like multiple questions we could have answered. Then like first question we chose was that so one of the long-standing puzzles in MAP kinase signaling is how plants process diverse signals, diverse stimuli through the same core MAP kinase cascade that are implicated in immunity and stress and also hormones. So as we all know, there are multiple, numerous actually individual studies have shown that core MAP kinase activation, for example, MPK3, MPK6, and MPK4 in Arabidopsis, the core MAP kinase activation to many different types of signals. There's activation of MAP kinase in response to many different types of signals, suggesting the involvement of MAP kinase for the signaling pathway. So then the question is, when we put them all together in the same context, the critical question is how in all these different signals processed through the same core MAP kinase, however, they still produce different cellular output. So we hypothesized that maybe different signals bring different combination of MAP kinase dynamics, that can contribute to the signal-specific responses. So here, when we say different combination of MAP kinase dynamics include three factors, I can say. For example, which MAP kinases are activated, either MAP kinase 6 and or 3 and or 4 or 3 together, and how long they are activated, the duration of the activation, and also how much activity, how strong the activation is. So to test this idea, we first carried out a systematic time series and quantitative measurement of MAP kinase activation program in a uniform experimental system with, here I listed 10 different signals. So we used 7 days old seedlings grown in a liquid medium of liquid MS medium. And then in these seedlings of a Y type, grown in liquid medium, we applied 10 different types of signals individually, comprising MEMS, microbe-associated molecular patterns, including FLAG22, alpha-18 or chitin, and also DAMP, damage-associated molecular patterns, so PEP1 and PEP3. These are the endogenous plant signal peptide to amplify dangerous signals, and also multiple stress signals, including mechanical wounding and UVB and hydrogen peroxide, and also plant hormones, including ABA and JA. So this is a Western blot with phospho-ERK antibody. So this antibody can detect specifically phosphorylated version of MAP kinase, so active version of MAP kinase. And you can find 3 bands here. So the top band indicates MPK6, phosphorylated MPK6, and middle band indicates phosphorylated MPK3, and this bottom band indicates phosphorylated MPK4. So as you can see, all these signals except for ABA and JA, these signals, they all triggered very rapid activation of MPK6, MPK3, or MPK4 and or MPK4 with different signatures. For example, immune signals such as FLAG22 and F18, they rapidly and strongly activated all three MAP kinase, with a longer duration for MPK6 and MPK3, but transient activation for MPK4. Whereas chitin and PEP1 and PEP3, they activated MPK6 and MPK3. But very moderate activation of MPK4. And remarkably, Wnt signal only transiently activated MPK6 and 3, but not MPK4. And UVB and hydrogen peroxide triggered strong and sustained activation of MPK6 and moderate activation of MPK3, and also very subtle activation of MPK4. So, and this is the quantification of activation profile of MPK6 and MPK3 and MPK4 by different signals based on this Western blot result. So collectively, we found that different MAP kinase dynamics between different signals. So we predicted that these are linked with, they must be linked with different transcriptome responses, presumably by regulating transcription factor activities. So to link these MAP kinase activation profile to transcriptome response in different types of signals, we applied a 3-MBIP, which is ATP analog inhibitor of MAP kinase, to these three different genotypes: wild type IMPK36 mutants and IMPK4 mutants, without or with different types of signal that differentially triggered the MAP kinase activation. And then we performed a comparative transcriptome analysis using RNA-seq after 1 hour of treatment to see how a signal response in wild type is differentially changed in IMPK mutants, to see the role of MAP kinase activity for the activation or repression of transcriptome in response to different types of signals. So from RNA-seq data analysis, we first identified stimuli response genes in wild type with these 7 different types of signal, FLAG22 and PEP3 as our immune signals, and mechanical wounding, UVB, hydrogen peroxide as a stress signal, and plant hormone and ABA and JA. And as you can see, the size of the gene response varies by individual signal. For example, immune signal, they really induce a huge gene changes, but compared to immune response, this immune signal, ABA or UVB, their signal response amount was actually smaller. So this suggests that systems like the plant's response is shaped signal specifically during the certain period of time. So these are all 1 hour treatment after signal. So in a given time, the amount of response is different by the given signals. So then after we identify response genes in wild type, and then we measured relative expression of those response genes in the inducible MPK36 mutants and inducible MPK4 mutants. So in this heat map, blue means that there is a less expression of the corresponding genes in the mutants, and yellow means that there are actually more expression of the corresponding genes in the mutants. and this white blank means that there is no difference between wild type and mutants. So strikingly, these are actually the summary of the result by showing a pie graph. So strikingly, stimuli response transcriptome has broadly changed in either of MAP kinase mutants, between different signals from 84% of the total transcriptome, total inducible transcriptome, or 5%, very small amount of the total transcriptome. And then the levels of each module dependency, for example, whether it's MPK36 module dependent or whether it's MPK4 module or dependent, they were signal specific. For example, the primary immune response genes stimulated by FLAG22 and PEP3, they were actually influenced by, so I think it's better to look at the heat map. So they were actually influenced by both MPK36 and MPK4, as we found all three MAP kinase were activated. However, the wound response, if you remember, we only found activation of MAP kinase 6 and MAP kinase 3. And also in transcriptome analysis, we found that about 50% of the wound response genes were differentially expressed in MAP kinase 3/6 mutants, but not in MAP kinase 4 mutants. They were, because as you can see, they are just like a blank color, indicating that the gene response is pretty much similar between wild type and MPK4 mutants. And then, so also like a similar case, UVB and hydrogen peroxide, we found like more genes are affected in MPK36 mutants, and a small amount of genes were affected in MPK4 mutants, which is linked with MAP kinase activation profile that we have performed previously. And then in case of ABA-JA, we couldn't detect any MAP kinase activation, at least in our experimental design. And also in this transcriptome analysis, we didn't find any much significant difference of gene expression between wild type and both of the MAP kinase mutants. So these comparative transcriptome analysis suggests that different signal mediated MAP kinase activation dynamics is linked with signal-specific transcriptome network. So then we combined all these stimuli response genes regulated by MAP kinase, and then we put them in the same dimension to identify a common MAP kinase regulated stimuli response genes. So in these network illustrations, so here the white balls indicates different types of signal, different types of stimuli, for example, PEP3 and FLAG22, ABA, UNI, JA, UVB, and hydrogen peroxide. And these colorful dots indicate stimuli responding genes regulated by MAP kinase in our transcriptome analysis. So the dots with one connection, for example, these purple dots, they have one connection to the ball. They are only inducible by this specific signal. So these are the signal-specific MAP kinase mediated responding genes. And dots with pink color, they are the MAP kinase mediated responding genes that we can find in two or three different stimuli. And dots with sky blue color, they are the ones with more than 4 connections, meaning that they are the core MAP kinase network response genes found in more than 4 different types of stimuli responses. So in this core MAP kinase network, when you look at the gene identities, include 269 genes composed of various signaling molecules such as peptides and kinases and phosphatases and calcium signaling components and various transcription vectors and immune sensors and enzymes for secondary metabolites, suggesting the roles of core MAP kinase network to mediate proper coping responses which are common to different types of stimuli. And then furthermore, when we look at the signal-specific MAP kinase network genes, which are these purple dots in these network illustrations, each of them are enriched with distinct biological process in different signals, suggesting that in addition to these core MAP kinase network, plants utilize specific MAP kinase mediated responding genes to have more tailored and more sophisticated defense response to the different types of stress. And our study suggests that these are driven by the different combination of MAP kinase dynamics upon signal perception. And then we focused on immunity. So now I'd like to further discuss the temporal dynamics of individual MAP kinase in a specific signaling, especially the immune response. As you can see here, using a FLAG22 as response as an example, so we found that 3 MAP kinase activation, very strong activation by FLAG22. However, this activation with distinct temporal activities. As you can see, MPK6 and MPK3, after very rapid activation, the activity is sustained. This actually up to 1 hour. And however, in case of MPK4, the activation is very transient. So then we actually performed the transcriptome analysis. And then this transcriptome analysis shows that about like 80% of the immune genes are regulated by either of MPK3/6 or MPK4 modules. So then the question was, this RNA-seq transcriptome analysis, which is a FLAG22 response that we see the different regulations in MPK3/6 and MPK4, whether we can link the transcriptome response to the different activation profile between MPK3 and 6 and MPK4. So this is a FLAG22 1-hour treatment. And I want to stress that there are actually substantial amount of genes in MAP kinase 3/6 mutants, which are shown as yellow color. So we actually expected to see that genes must be repressed. I mean, the immune genes, most of the immune genes will be repressed, like these blue colors in both of the MAP kinase mutants, because we thought that MAP kinase, they function as a positive regulator of the immune gene activation. However, we found substantial amount of genes are actually more activated in MPK3/6 mutants, indicating that for these genes, actually MPK3/6, they function as a repressor. So then we suspected that maybe this could be some kind of feedback repression of MPK3/6, and this is 1 hour. So we looked at earlier time point and then combined the data set. So this is the combined data set of 30-minute and 60-minute FLAG22 response only in wild type, and I defined activated genes, only immune gene activation, so these red colored genes in this heat map, into 3 groups. So type 1 meaning that 30-minute specific genes, and type 2 meaning that 30-minute and 60-minute responding genes, and type 3 genes are the 60-minute specific genes. And then in each type of the genes, we performed comparative analysis of transcriptome to learn how these expressions are regulated in MPK3/6 and MPK4 mutants. So based on the regulation dynamics, we found something very interesting. In case of type 1, so 30-minute specific genes, we found these genes are repressed or not regulated in both of the MPK36 and MPK4 gene. And when we actually looked at the type 2 gene, which are begin to express at 30 minutes and then stayed up to 60 minutes, we found very interesting dynamics of regulation patterns in MPK36 mutants. As you can see, at 30-minute time point, most of the genes were repressed, right? However, in 60-minute time points, we begin to see that some genes, they were not regulated by MPK36. They begin to have more induction in MPK36 mutants, and the genes that were not that was repressed by MPK36 in the mutants, their repression is released in the later time points. And very interestingly, these set of genes showing different dynamics of regulation, they were tightly linked with the genes that are differentially regulated in MPK4 mutants. So then from this transcriptome analysis, we hypothesized that maybe MAP kinase activity, if you remember, here the MAP kinase 4 activation is very rapidly reduced, and this reduction of activation is mediated by MPK 3 and 6. So the earlier time point, all three of them are activated, but later time point, the MPK 3 and MPK 6 activity, it is still remained, and this activity repressed MPK 4. So that's why in MPK 3/6 mutant, the activity is gone. So then MPK4 activity is still sustained in the mutant. So then that's because the genes that are regulated by MPK4 is more highly activated in MPK3/6 mutants. So based on the regulation dynamics in different MAP kinase mutants, between MPK3/6 and MPK4, we found 6 categories of the immune genes. So category 1 genes are regulated by only by MPK3/6 module. They include known immune signal amplifiers and receptors and transcription factors for secondary metabolite with antimicrobial functions. And category 2 genes, and these are the genes regulated by MPK4. They also include prominent immune players. And as I explained, at later time point, MPK4 activity is feedback repressed by MPK3/6. That's why in MPK3/6 mutant condition, there is a superactivation of MPK4. So these immune genes are more highly activated in MPK3/6 mutants. And category 3 genes, we found that these genes are regulated by both MPK3/6 and MPK4 pathway, and category 4 genes, and they are regulated by MPK3/6 and other components that are also repressed by MPK3/6. In category 5, they are actually regulated by other components, but then the activities of other components are repressed by MPK3/6. And we found 20% of the total immune genes, they are not dependent on any of the MAP kinase pathway. So I want to make a brief summary. So in summary, after immune signal sensing, activated MAP kinase cascade regulate transcription of 5 different categories of immune genes involved in immune sensing and signaling and network rewiring by modulating transcription factors amount and antimicrobial metabolite synthesis. And this regulation is mediated by different dynamics of MAP kinase. For example, at the earlier time point, they all strongly activated and induce these genes. however, later time point, MPK36, they are actually dual function kinase, so they can actually repress the activity of MPK4 and other components. And although I'm not showing today, it also repress its own activity. So, suggesting that the role of MPK36 not only as an immune activator, but also they function as the molecule, the feedback repressor that can dampen the signal response, because a hyperimmune response is also not good for organism. So in our study, we proposed a new function of MPK4, which was difficult to study their roles in innate immunity. So we found that substantial amount of the immune genes are regulated by MPK4, and we were able to define a set of genes that are regulated by specific MAP kinase cascade. And these dynamics in the regulation, not only just the FLAG22, not only just the MEM signal, we also compared this response against them, PEP3 or bacteria protease, and then we all find similar regulation patterns. So meaning that this MAP kinase regulation is conserved in broad immune signaling pathways. So to make a very long story short, I and my former colleagues, we were able to propose diverse functions, definitive functions of MAP kinase in multiple cellular programs. Although I didn't bring everything today. So I talked about stress and immunity response, but we also found the role of MPK36 in cell type patterning, and also a role of MPK4 in cytokinesis and triggered immunity, and also suppression of the autoimmunity that we can phenotype by different concentration of the chemical inhibitor. So then here at GUGC, here at Ghent University Global Campus, in my lab, I addressed another question, the next question, which is, now we know that now we have a tool to define the signaling pathways that are regulated by MAP kinase 36 or MAP kinase 4. And then the question is, what are the molecular details in each specific signaling pathway? For example, MPK4 regulates cytokinesis. Then what are the downstream signaling components in the regulation of cytokinesis by MPK4? Or what are the downstream components of MPK36 to regulate cell type patterning, or something like that? So one of the phenotype of MPK4, so when you apply the chemical inhibitor, you can see that the root growth is very largely defected, because MPK4 is involved in the regulation of cytokinesis. And as you know, like cytokinesis is the last step of cell division. So if we apply a 3-MBIP ATP inhibitor, you can see this very dynamic dramatic defects in the root growth of MPK4 mutants. That's because of the defects in cytokinesis. So as you can see, in this 5 macromolar and 10 macromolar of the inhibitor condition, cell division is not complete. So the plants cannot grow because they do not finish cell division. So, and also it's been known that MPK4 activity is important for cytokinesis when people study genetic mutant of MPK4. So we know that MPK4 regulates cytokinesis. However, the detailed molecular mechanism by which it regulates the process, the mechanism is largely unknown. So in my lab, we addressed a fundamental question that how MPK4 regulates cytokinesis. What's the molecular details in this MPK4 pathway to regulate cytokinesis? So to answer this question, we combined our chemical genetics approach with a classical forward genetics approach using EMS. So as you know, EMS generates random mutations in the genome by changing GC pair to AT pair. So after generating random mutations in the genome of the chemical inducible mutants, chemical inducible MPK4 or MPK36 mutants, and then we performed phenotype-based suppressor screening after applying ATP analog 3-MBIP. So if you remember, if we apply 3-MBIP, the mutants have a very short root phenotype, but then we try to find the seedlings with long root phenotype. So here is the workflow. So this is the phenotype that I just explained. If we apply 3-MBIP, the root length is very short. So after generating EMS mutagenized line of IMPK4 or IMPK36 at M2 generation at seedling stage, we look for the ones with long root phenotype, and then we confirm. So from this screening, we found about like 50 the mutants that were showing the white type like phenotype. So and then in M3 generation, we confirmed that 47 out of 50, they reproduced the phenotype, meaning that the phenotype was stably inherited to the next generation. And then we did backcross to the parental line to IMPK parental mutants to generate a mapping population. So then in this BC1F2 segregating mapping population, we determine the inheritance pattern, whether it's recessive or dominant, and then we also perform the whole genome resequencing to identify the causal mutations associated with a gene. So here I'm showing an example of one of our suppressor mutants of IMPK4. So we name it as SIMPK4, suppressor of IMPK4. So you can see that with the DMSO there is no difference. However, in the treatment of 3-MBIP, IMPK4 mutants have very short root phenotype because they have a defect in cytokinesis. However, in SIMPK4 suppressor mutants, their root lengths root growth is almost recovered back to the level of wild type. So indicating that whatever the mutation is, the gene product is the downstream of MPK4 and then regulate the cytokinesis. So upon the developmental cue for cell division for cytokinesis, there is a MAP kinase activations, and this MAP kinase activity somehow the activity of our SIMPK4 gene product, and this gene product regulates cytokinesis. So then when we have the mutation on these genes, without MPK4 activity, the plants can perform cytokinesis with no problem. So then when we find this SIMPK4 mutants, we were curious that whether this SIMPK4 gene product is only involved in cytokinesis or also involved in other pathway of MPK4, such as immune pathway, because as I mentioned, MPK4 is also function as an activator of immune response genes. So we looked at the immune response genes activation in wild type and IMPK4 and SIMPK4 mutant. We looked at two immune genes, and as you can see, after FLAG22 treatment, there is an activation of the immune genes in wild type, but no activation of these genes in MPK4 mutants, and also no activation of these immune genes in SIMPK4. So meaning that SIMPK4 gene product is only specifically involved in cytokinesis pathway, but not in the immune pathway. So collectively, this result suggests that we have successfully identified downstream components that are specifically involved in cytokinesis. So identification of SIMPK4, now we have about like 40 mutants, will provide hopefully a comprehensive overview on MPK4 cytokinesis regulatory network. And we also apply the same approach on MPK36 pathway, and we are looking for, searching for the mutants that are suppressor of MPK36 mutants. So hopefully we can also find previously unrecognized downstream factors in MPK36 pathway involved in cell differentiation or stress response or immunity. So we can further dissect the signaling pathways at the molecular level. So, so our mission is in my lab is to understand plant resilience at the molecular level and contribute to have the knowledge to increase crop yield. So as I mentioned, MPK4 MAP kinase is involved in multiple pathways, including immunity, stress response, and development. And in our suppressor screening, we can find some candidate genes that are downstream of MAP kinase 4, but only specifically regulated, involved in cytokinesis, not in immunity. So hopefully we can, using this knowledge, after knowing the identity of the genes and then knowing the molecular link between the gene and MAP kinase, we can have the knowledge of how plants have resilience, between for the balance between growth and development and immunity and stress response. So we are at the stage of here, I guess, in the between here and here. So after a suppressor screening, we have several candidates, and then more than half of them we are finishing up the whole genome resequencings, and then we are trying to identify the molecular link between the MAP kinase and our target gene product to understand the mechanism. Okay, so that's it for today. So in my group, I have three very talented and dedicated PhD students, and one research professor just to join the group, and I also work together with three GUGC, Ghent University Global Campus Bachelor students. And this is the lab picture when we had a lot of visitors. Not all of them are staying in the lab. We are also collaborating with Professor De Jager's lab at VIB in Belgium, and also collaborating together with Human Genomics lab at Hanyang University in Korea for SMP analysis. And the work that I presented today is supported by GUGC Internal Research Fund and also Ghent Startup Grant. And also, I haven't talked about it today. Some of the immunity research part has been funded by NRF Basic Research Fund. Okay, so thank you very much for listening. And if you have any questions, because this is an online video of seminar, so if you have any questions, please contact me through email. Okay, thank you very much.